great. And so with that, I will introduce our speaker for today. So Dr. Richard Sherwood is an assistant professor of medicine at Harvard Medical School. He is originally from New York and attended Stanford University for his undergraduate studies. Dr. Sherwood made his way back to the East Coast, moving to Cambridge to start his PhD at Harvard University. Dr. Sherwood worked on embryonic development and specifically endodermal organ specification under Doug Melton. And after receiving his PhD, he made an even bigger move across the river to Boston, where he performed a fellowship at Brigham and Women's Hospital and Harvard Medical School. So now Dr. Sherwood runs his own lab at Brigham and Women's Hospital and Harvard Medical School, and he and his team focus on developing creative technologies that combine stem cell biology, CRISPR-Cas9, genome editing screens, and computational biology to understand gene function and regulation. So please join me in welcoming Dr. Richard Sherwood. I'm going to turn it over to you.
Okay, great. Thanks a lot, Jessica. So I am going to share some work on basically how our lab, which focuses on genetics and CRISPR-Cas9 genome editing, how we use molecular cloning. I guess I'll let Jessica interrupt. I'm happy to take questions if anything's unclear or if you want more details on anything in the middle, or wait until the end, but I'll let Jessica handle that. So I guess in terms of the purview of my talk, I'm going to start with some basics of gene cloning, but very, very little on that, and then just go on to kind of how our lab uses these techniques, because I think that's kind of what the goal of today's talk is. So just starting out, I just wanted to give some kind of get everyone on the same page in terms of the basics of cloning. So kind of the absolute basics that are kind of the textbook version of molecular cloning that have been around since the late 70s, early 1980s, is this idea of restriction enzyme cloning, where you take these enzymes that are purified from bacteria that cut very specific DNA sequences. You have a donor plasmid where you cut with a set of restriction enzymes that leave usually sticky single-stranded ends. And then you have a recipient plasmid where you basically use those same sticky ends and insert whatever the insert you want from one plasmid to the other. So this is sometimes called subcloning. And usually then this is done by a restriction enzyme digest of both plasmids, followed by a DNA ligase. So this is not a form that we pretty much use at all in our lab, because it's just been supplanted by more updated techniques. One update that's worth mentioning is that basically if you don't have this perfect setup where you have a donor plasmid or recipient plasmid with the exact same restriction enzyme sites, one kind of cheat is to still use these restriction enzymes on the recipient plasmid to basically cleave it open, but then to basically add those restriction enzyme sites on your insert through the polymerase chain reaction, or PCR. So the way that is done is you have a primer sequence, which will allow you to specifically amplify whatever you want. And then you add through a tail on that sequence that is not complementary to the sequence in your donor plasmid, but has this restriction enzyme site on it. And you do that with the other restriction enzyme site on the 3' end. And then basically what you're left with is a double-stranded fragment that has each of these restriction enzyme sites on the ends that you can then also digest with the appropriate restriction enzymes and insert into the recipient plasmid. And so this kind of gets around the fact of needing these restriction enzyme sites to be on both the donor and recipient plasmid. Now, actually, again, we don't use this technique either in our lab. We use basically a kind of more updated technique that involves this principle of PCR amplification of the insert, but actually even goes one step further, which basically just makes the reaction more efficient. And that step is to use what's called Gibson assembly, or the version we use is called AnyBuilder, which is just kind of how the manufacturer, New England Biolabs, which is kind of one of the main vendors of restriction enzymes and cloning reagents, has dubbed it. But basically the difference here, so the part where you have the recipient plasmid that is linearized, usually using restriction enzyme sites, is retained. However, when you PCR amplify the part that you want to add in, or the donor or insert, you can actually, you don't even need to put restriction enzyme sites. All you need to do is put an overlap, a sequence overlap with the recipient plasmid. Typically with these reagents, you use about a 20 base pair overlap. So you'd have whatever your insert is, and then you have two primers, your forward primer and your reverse primer, which basically then add 20 base pairs of exact overlap with the sequences in your recipient plasmid. Now, the cool thing about these reagents is that they actually work within a sequence range. So if you basically want to get rid of a restriction enzyme site, you can basically just put the 20 base pairs of homology with the recipient plasmid a little upstream of the restriction enzyme sites you use to cut. And when it comes together, those restriction enzyme sites will be gone. Or you can add a site in between your insert and the plasmid just by, same thing, using PCR primers to basically add a restriction enzyme site, or maybe a tag onto your, if it's a gene of interest, or add a mutation. Is a common way that we basically will do add mutations to plasmids. And then you use this Gibson assembly technique, which involves using an exonuclease to chew back the 5' end on both the insert and the recipient plasmid, and then use this combination of a polymerase and a ligase to basically paste it together. And this is all actually just done in a single reaction. So this is this Gibson master mix, or any builder master mix. So I guess the reason why we use this in the lab is it tends to be more efficient. The overlap, as mentioned, between the insert and the recipient plasmid is typically on the range of 20 base pairs or longer, which just will have much higher homology, or sorry, much higher annealing temperature than the ends of a restriction enzyme site, which might only be 2 to 4 base pairs long. And it just gives this flexibility for new students in the lab. It's great because really, as long as you kind of know the eventual sequence you want, you can just, it's very easy to program in just by adding inserts, or sorry, adding tails onto your PCR primers, the exact sequence you want on both ends. There's actually another trick here which can make this even easier. So still, this procedure relies on having restriction enzyme sites A and B to cut open your plasmid. However, often actually these days, you can actually get rid of that step even by just taking a plasmid and basically just doing a circular PCR reaction. So basically if you just place, so it's actually, that's depicted here, your vector primers, you can actually just make outward facing primers that basically amplify the entire vector all the way around, with the exception of this part where you want to add a sequence. And then you have your primers for your insert that basically just overlap by 20 base pairs. So you can basically get rid of the entire use of restriction enzymes and just acquire whatever plasmid sequence you want. So this, again, is just the bread and butter. This is the only technique that we use in our lab, with kind of those variations I talked about of, if possible, we tend to use restriction enzymes because the downside of PCR is it does have an error rate. So if you use this method of kind of round the world PCR for your vector, you end up having to do more Sanger sequencing verification to make sure you didn't insert some sort of mistake, especially since a lot of our donor plasmids are on the order of 10 to 20 kb long. And then just kind of following up with the basics, how do we verify that we got the right sequence? Because no matter what you do, if you do restriction enzyme of your vector, sometimes there is kind of remnant circular plasmid left after you cut out your gel band. or at some rate, maybe you can get recombinations using Gibson assembly, where basically the homology, where it uses a different homology because the exonuclease can chew back such a long region. You can actually kind of get deletions depending on your strategy. So regardless, we always use these two steps to verify the plasmid. One is diagnostic digest, where we come up with restriction enzyme sites that should give a unique banding pattern if the insert was added correctly, as opposed to if usually our kind of comparison is if there was no insert or if it was the original donor plasmid. And then we always use Sanger sequencing to verify the plasmid identity, because again, especially with these Gibson assembly techniques that are more PCR driven than the traditional techniques, it's always possible to get short single nucleotide variants or indels in your plasmid, which can mess up the function without being easy to see using a restriction digest. So that's kind of the basics of cloning that we use in our lab. And so now I'm just going to kind of move forward to how we use cloning in our lab in general, and then go on to some specific examples. So in terms of in general, we're a genetics lab. So basically the two things we tend to do are want to make cells express more of a gene or less of a gene. So in terms of expressing more of a gene, we often use a system called the RTTA, or reverse tetracycline transactivator system, where basically there's a molecule called RTTA that basically, in the presence of a small molecule called tetracycline, or often there's a more stable derivative called doxycycline that we use, in that presence of that molecule, it will bind to a very specific DNA sequence called a TET responsive element, or TRE. And so what this allows us to do is we can insert a gene into a cell, and when we add this tetracycline or doxycycline chemical, the cell will turn on the expression of that gene. Sometimes if we don't need that gene to kind of be inducible, we'll replace this TET responsive element promoter with a constitutive promoter like the CAGs, CBA, or EF1 alpha, or PGK. Those are kind of common mammalian expression promoters, where when we introduce a plasmid in the cell that has a promoter and gene of interest, the cell will constitutively pump out copies of that gene. Often we try to insert these into the genome of our cells. So that would be, I guess, not depicted here. Somewhere else in our plasmid, we'll either have a lentiviral LTRs, which basically allow the lentiviral system to flip this randomly into the genome of cells, anything that's basically in between the two LTRs of the lentivirus. Or alternatively, we use transposons, which basically, same thing. If you add a transposase to a cell along with a plasmid that has an insert flanked between the transposon recognition sequences, it will also be randomly flipped into the genome. And that allows us to basically get more stable expression of whatever gene in our cells. And often inside either those transposon or LTR sequences, we'll put a separate cassette which has resistance against a mammalian selective agent. The most common ones we use are blastocidin, hygromycin, puromycin, or neomycin. And basically those are all drugs that will kill mammalian cells unless there is a resistance gene. So usually what that cassette is basically a constitutive promoter driving expression of that resistance gene. And in the presence of that, any cell that has the entire cassette, which includes this inducible or constitutive transgene, will stay alive. So I hope that makes some sense. I kind of tried to spit out some technical terms, mainly just because those are specifics that probably ring a bell to a lot of people. But the general concept is we often clone genes in a setup that either has a inducible or a constitutive promoter that allows us to basically turn on that gene in cells. On the other hand, we do a lot of gene knockouts, where basically the state of the art now is using CRISPR-Cas9. And so for that, basically the CRISPR-Cas9 system, which I guess you'll hear more about next week, has two components. One is an enzyme, a Cas9 enzyme, and the other is a guide RNA, which is a small RNA species that has a fixed hairpin sequence that will basically bind to the Cas9 protein. And at the front of that is what's called a spacer sequence that will basically direct the complex of Cas9 and this guide RNA to a specific region of the genome that's basically guided by usually 20 base pair spacer sequence that is corresponding to that region in the genome. So in terms of the specifics of cloning that, the two components are, one is a guide RNA, where we typically use a U6 promoter, which is transcribed by polymerase 3, which basically allows the transcript to stay in the nucleus where it needs to be used for Cas9, because we tend to be messing with nuclear genes, whereas most transcripts with normal promoters or polymerase 2 promoters get exported to the cytoplasm. The second fixed component is this guide RNA scaffold, which is again the hairpin that will, when this gets transcribed into RNA, this is a hairpin that will complex with Cas9. And often our plasmids, similar to other plasmids, will have some sort of lentivirus or transposon sequences and drug resistance flanking them. The variable part, which is what we do a lot of cloning of in our lab, is what that spacer sequence is, because we can basically direct Cas9 to any place in the genome. We often basically clone in guide RNAs into this spot that's in this picture, just flanked by 2 restriction enzyme sites. So again, using the techniques described before, we cut this vector with restriction enzymes and then insert, using our Gibson assembly technique, a spacer sequence that's flanked with 20 or more base pairs of homology on either side, which then will basically get seamlessly inserted into this vector. Then we have our Cas9 expression. This is just a version, actually, that this version is called lenti-CRISPR v2. It's a lentiviral version where there's actually an Cas9 enzyme as well as a U6 guide RNA cassette in the same plasmid. So this is kind of an all-in-one system where you can both express your Cas9 and your guide RNA in the same plasmid. For our lab, we clone a lot of different versions of Cas9. There's Cas9 from different species that recognize different, that basically allow you to recognize different sequences in the genome. Again, you'll hear more about this next week, but just to give a very slight overview, in addition to this 20 base pair spacer sequence, you need to have in the genome what's called a PAM sequence, which is specific to each Cas9 species. So if we want to target different regions of the genome, we might want to change the PAM sequence. Also, there's different versions of Cas9 editors. There's what's called dead Cas9, where instead of cleaving the genome, you can basically cause things to bind to the genome, or cause the Cas9 enzyme to bind to the genome without cutting. And then there's kind of versions of Cas9 that basically either are more or less specific or will allow you to do different things with the sequence. I'll go over that a little bit at the end, but basically we do a lot of cloning of swapping around what version of Cas9 is in a plasmid like this. Then one other component that's not directly cloning, but is essential to all our cloning efforts in the lab, is most of our lab, we work on high throughput approaches where we don't want to just clone one thing at a time. We want to clone thousands of things at a time, whether they're thousands of guide RNAs or thousands of plasmids. To do that, there's companies, one is called Twist Biosciences, and there's several others that basically will clone oligonucleotides at scale. Without getting too technical, the way they do this is they basically attach the beginnings of an oligonucleotide to a solid support, and then they undergo a series of chemical reactions that basically kind of build up base by base a DNA sequence, and they can actually do this on a chip up to really an arbitrary number on the order of 100,000 different oligonucleotide sequences, up to 300 base pairs currently is the limit. And so what we do with those is, and I guess that's shown here, is we often make, for example, CRISPR libraries where we want to clone thousands of guide RNAs in a pool. This is often called library screening, I guess because each guide RNA is kind of like a book, a different book in a library. And so what we would order from this oligonucleotide synthesis company is a set of thousands of guide RNA spacer sequences flanked by a common set of PCR primers. And then that would feed into our Gibson assembly system, where we just take this entire pool, and because every single oligo in this pool will have the same forward and reverse PCR primers, that allows us to clone them in a pool into a fixed vector. And so now, instead of just cloning one thing at a time, we have this pool of vectors where each one will be exactly the same, with the exception of this 20 base pair spacer sequence, which can be any number of thousands of different versions. Because those thousands of versions will direct Cas9 to different places in the genome, we can then basically infect these in cells in a pooled fashion, knowing that each cell will get a different spacer sequence, which will in turn lead to Cas9 breaking a different gene, for example, and that allows us to do these high-throughput genetic screens. Actually, that's going to be the first example I talk about. So now I'm going to move on, kind of after talking about general techniques we use for cloning, some specific projects that in our lab that use these kind of advanced cloning techniques.
So Rich, can I interrupt you before you dive into these projects? We have a couple of questions in the chat.
Sure.
So the first question is, what are the bioinformatic websites used to determine base sequences? UniProt, UniParc, NCBI, UniRef, if you want to speak to that.
Sure. So specifically for CRISPR-Cas9 experiments, there's a whole host of databases that we make use of. I guess the basics are, if you want to target a gene, you just need to find a 20 base pair sequence that is unique to that gene, that then also has a valid PAM sequence, which is this kind of 2 to 3 nucleotide sequence that follows the guide RNA or the spacer sequence, and is unique to each Cas9 species. So now there's been a lot of tools that basically identify unique sequences to for each that might be unique to each gene, and they really, I think maybe I could follow up after on the specific links to them, but there's kind of a whole host of them that have different features of what you want to do. I'll talk about one that our lab has created in a few slides. But so for CRISPR, basically the main thing is identifying valid target sequences in the genome. And then I guess, really it depends on the project. So if we're looking for gene sequences, so we often will use a site like UniProbe or NCBI BLAST to identify or a UCSC genome browser is another one we use all the time, since that's basically just has the complete genome sequences of any organism. So if you want to find a gene, the genomic DNA or cDNA of that, that's a great resource.
Great, thank you. And we have one other question here, which you may want to address now, you may want to save, we'll see. So what are other cloning methods, and what are the advantages of cloning with CRISPR-Cas9 over other methods?
Sure, so there are certainly other cloning methods. So as mentioned, kind of really the goal is to put 2 pieces together or multiple pieces if you can. With this Gibson assembly method, you can actually combine up to somewhere on the order of 5 to 10 different sequences in the same reaction, as long as each one has a unique 20 base pair overlap with the previous one. So you can kind of string different sequences together, and it really does work. So, you know, we've put 5 different sequences together, and as long as they have these unique 20 base pair overlaps, we can basically seamlessly get them all in the same plasmid. There's another technique called user cloning, which basically is an alternative kind of new technique similar to the Gibson assembly technique, that instead of requiring this 20 base pair overlap, it involves this very specific uracil sequence that shouldn't exist in DNA, and that there's a set of enzymes that basically specifically recognize that. So that's, I know, one that other labs use instead of Gibson assembly.
Perfect. Thank you so much for answering those. We'll look forward to hearing now about your projects.
All right. So again, these are just a few examples of projects that use cloning in our lab. One that's, I guess, kind of timely to the moment is we wanted to do a screen of genes that affect the toxicity of certain SARS-CoV-2 drugs. And specifically, we focused on 2 drugs, remdesivir and hydroxychloroquine, since they're very commonly used now in patients. So what we did is we basically developed a genome-wide targeting CRISPR lentiviral library. So what this now means is that we took every gene in the human genome, and we actually designed 4 spacer sequences that you should uniquely target each gene. That comes out to about 80,000 different guide RNAs. We had them synthesized in one pool, cloned them into a lentiviral library that includes Cas9 and that guide RNA sequence, and then infected them into a pool of cells such that, again, every cell should express a different guide RNA, which means it should get a different mutation in its genome. And the cool thing about this sort of approach is that we can then afterwards figure out, for example, if there's a guide RNA that specifically kills cells when you give them hydroxychloroquine, then that guide RNA should drop out of the population when you dose the cells with hydroxychloroquine. So then we can basically just use next generation sequencing, which is basically kind of high throughput Sanger sequencing, where you basically just can figure out how many times each guide RNA or each particular spacer sequence shows up in your pool of cells to see which spacer sequences show up more often or less often upon dosing of a drug. So this is just kind of an example that I will not go over in detail of the sort of data that we get out. Basically, this is on the x-axis here, we're looking at, on the right, are genes where when we add hydroxychloroquine, the guide RNAs are more abundant than when we add a control chemical, which is just DMSO, which is an inert chemical. And on the left here are genes that basically are less frequent in the population, or where spacers to those genes are less frequent in the population when we've added hydroxychloroquine. So basically, these are, on the left, are genes that in the presence of hydroxychloroquine, lacking that gene is toxic. And on the right here are genes that when you lack that gene, hydroxychloroquine is no longer toxic. And so this set here are very interesting candidates for us for reducing the toxicity of a drug treatment. And so what we can do with this extremely powerful technique, where we can basically screen through what happens when you lose the function of every single gene in the genome, is we can then kind of try to group them into biological pathways. And we can find, for example, that when you disrupt peroxisomal membrane transport, cells are no longer cells are no longer sensitive to hydroxychloroquine. But when you disrupt endocytic recycling, cells are more sensitive to hydroxychloroquine. And then we can use biological follow-ups. This is showing, just validating one hit that we found here, where which was surprising to us, that when we add hydroxychloroquine, it actually disrupts the uptake of LDL cholesterol in cells I should say hydroxychloroquine, that basically gets stuck in these vesicles because there's a defect in endocytic recycling. So again, the point of this is not for you to understand all of this biology, but just to kind of display that using this technique of library cloning of CRISPR guide RNAs through Gibson assembly, we can basically very easily, in the course of 2 months, dissect the pathways that are used in the cell for drug toxicity. A second project that I'll talk about again briefly, just because it kind of required an even more complex version of cloning, is we were interested in this idea that when you cut the cell with CRISPR-Cas9, the cell actually doesn't respond randomly in terms of how it repairs that damage. It seems from previous data that replicate after replicate, you get the same deletions and insertions for each site in the genome you target. However, that had only been done in a small number of sites. So we said, well, can we figure out a way to use CRISPR to target thousands of sites at a time and figure out what happens? So basically what we wanted to do is have an approach where we had a variable CRISPR-Cas9 guide RNA, but also pair it in the same plasmid with its target site. So this would be a site that has the exact recognition sequence of CRISPR in it. And what that would allow us to do is, in a very simple assay, figure out what sort of mutations CRISPR causes at thousands of different target sites. To do this, we needed to solve a problem, which was, how do you pair a guide RNA and a target site in an oligonucleotide where there's a sequence in the middle, which includes the guide RNA hairpin and a linker sequence, which is too large to encode in an oligonucleotide. An oligonucleotide, as I mentioned, can be a maximum of 300 base pairs. And so we couldn't encode both the guide RNA and the target site and everything in between in a single oligo. What we did instead is, because we still wanted to have the guide RNA and the target site linked, we made an oligo library where that guide RNA and the target site were simply separated by a restriction enzyme site called SSP1. And so when we amplified the oligonucleotide, we could then cut it with this SSP1 enzyme, and using a Gibson assembly step, we could actually insert the region that we wanted in the middle here, and then break it open again, such that basically we could then amplify this entire linear piece that now included the fixed chunk in the middle, and pop that into a vector. So this basically, again, required Gibson assembly at 2 steps. First, now we did an initial Gibson assembly that was able to basically insert a common linker sequence, and then a second Gibson assembly, which was then to insert this entire linear piece into a backbone vector that had other features like these transposon sites that allowed us to integrate into the genome. So again, same set of techniques, Gibson assembly, but kind of using it in fancy ways to allow us to basically solve a technical challenge that we couldn't have done otherwise. Without, again, going into detail, this allowed us to figure out that there's this very specific pattern of mutation that happens with CRISPR-Cas9. And because we had thousands of examples of it, we could figure out these cool trends, like basically when CRISPR-Cas9 cuts, often it creates an inserted base, and that inserted base was most likely to be a duplication of a very particular base that we could predict. And we could use this to make an algorithm where now, with this algorithm, you can basically type in any target sequence in the genome of mouse or human, and we will tell you what sort of mutations would you'd get if you chose a guide RNA that targeted that sequence. We'd tell you, in this case, you'd get a lot of 1 base pair insertions, you'd get a lot of 8 base pair and 11 base pair deletions, but not a lot in the middle. And so this is an extremely powerful tool that basically was only possible because of this advanced cloning. We've now moved on to kind of do other versions of this kind of two-step Gibson assembly process, where now we're actually testing both splicing and CRISPR-Cas9 in the same assay, where now we have a fixed guide RNA and a fixed target sequence, but that target sequence is actually in the context of a splicing reporter system that allows us to ask the question of what happens when you use CRISPR-Cas9 to disrupt a splice site. So again, having these abilities to do fancy cloning has really enabled us to ask questions that people couldn't ask before. This is another project which I think is another cool illustration of kind of the frontiers of molecular cloning. In this project, we actually wanted to ask what happens when you attach peptides to a version of Cas9 that I talked about that is called dCas9 or dead Cas9 that's used to upregulate and downregulate genes. So what we did is we attached a set of 18,000 peptides to dCas9 and asked which ones of them, when attached to dCas9, would upregulate gene expression, which ones wouldn't do anything, and which ones would downregulate gene expression. To do this, we needed to encode these peptides in an oligonucleotide library. But because now we can clone libraries, or we can design libraries up to 300 nucleotides, we could actually encode 85 amino acids worth of peptide in an oligonucleotide, flank it with fixed primer sequences, and clone it into this plasmid sequence where basically all other components are fixed. And so now the only thing that's different about these 18,000 plasmids that we've cloned in one pool is what peptide is attached to dCas9. And what we could find is that there were certain peptides that when we attached them to this dead Cas9 could strongly upregulate gene expression. So those could be used as basically tools to turn genes on or off. And this is extremely useful for thinking about therapeutic treatment for genetic diseases where you have too little or too much expression of a gene. And again, this was made possible by this idea that now you can clone oligonucleotide libraries that are that basically can encode for a peptide sequence and allow you to screen through thousands of different peptide sequences for activity. So I know this is probably a lot, and I'm not explaining the science behind these projects, but I just kind of wanted to give you a flavor of what cloning can do. So just to kind of give some final perspectives, the uses of cloning are really only expanding based on this idea that you can do these high throughput oligonucleotide libraries. One extremely promising use is in making antibody sequences. So at this point, most of the top selling drugs in the world are antibodies. And those antibodies are usually designed using a process called phage display, where basically on the order of 10 to the 10th or 10 to the 20th, which is an extremely large number of antibodies are made through oligonucleotide library synthesis, followed by cloning into a vector where basically the variable sequence of an antibody is encoded by these oligonucleotides. And so now you can screen through trillions of antibodies that have just one sequence different about them, and figure out which ones bind to your protein or your antigen of interest. I'm not going to go over this in detail, but basically there's a whole process worked out that is how a lot of the antibodies that are now kind of multi-billion-dollar drugs were created through the use of oligonucleotide library cloning, mostly using the Gibson assembly process to basically kind of pop them into specific places in a vector. This idea of oligonucleotide synthesis As I said, now we can clone on the order of thousands of sequences up to 300 base pairs. But this process of oligonucleotide cloning is really expanding quickly. And I think one exciting change in the field of cloning will be the ability to synthesize more and more complex things. I mean, we're really not far from the point where instead of now cloning peptides, we'll be cloning thousands of genes and soon cloning thousands of genomes. And you can imagine the sort of advances that could occur in science as the ability to synthesize and clone larger and more complex sequences. Lastly, and maybe I'll skip this part, or I'll skip some of this part, is that there's an alternative now. CRISPR-Cas9 has gotten so good that now a lot of the things that we used to think of doing using molecular cloning, for example, inserting a gene or a mutation into the genome, we can now actually do with CRISPR-Cas9. I will skip this part just to talk about kind of the best and brightest new tool in CRISPR, where basically it's called CRISPR prime editing. Please, if anyone's interested, I'm not going to do it justice, so you should look up the paper. But basically what you can do with CRISPR prime editing is you can actually specify any mutation that you want to introduce into the genome, from an 80 base pair deletion to a 44 base pair insertion to any form of change in between. So you can create a single nucleotide variant, you can add a sequence up to 44 base pairs or delete a sequence up to 80 base pairs, anywhere you want in the genome of any cell. And by just designing the right CRISPR guide RNA, again, using molecular cloning, cloning it into a plasmid, adding that to a cell, you can actually make the cell do the hard work and actually change the genome sequence in a very precise and predictable way. And it's not perfect yet. Still, when you do this, that kind of perfect mutation that you've programmed only occurs in maybe 10% of cells. But it kind of opens up these exciting possibilities where now, if you want to change the genome, instead of kind of having to go through this process of adding a whole gene to the genome, you can actually go in there and natively tinker with genomic sequence using CRISPR. So I think that's, again, going to be a really exciting development that kind of changes what we think of as molecular cloning, where now you can actually kind of directly do the cloning inside living cells. So that was all I had prepared. I'd like to just briefly kind of show some of the sponsors and people in my lab who did some of the work I talked about. And now I'd be happy to field questions.
Wonderful, thank you. So we have some general and some specific questions. So perhaps we'll talk about one practical question here. Where can we learn more? Where can we learn more about this? Any books or papers that you would recommend specifically? Yeah,
unfortunately, I'm more in the research field, not in the teaching field. So I think that's probably something that you guys at LabXchange are probably better equipped to answer. There's tons of academic review articles, probably fewer books, although there are a few books on the history and development of CRISPR. But I don't want to point out any specific examples because that's not really my expertise.
Perfectly fair. I will put a link in the chat in just a moment to our CRISPR cluster for folks who want to learn more on LabXchange. Another question for you, can you talk a little bit about how your lab is using CRISPR screening in response to COVID-19?
Sure. So, I mean, I very briefly talked about this project. So, again, we're more of a genetics lab. So we're not a virology lab, and we're not an immunology lab. And I would say those are kind of the two expertises that are most useful in understanding and figuring out treatments for COVID-19. The screen that I talked about was basically trying to understand how these frontline drugs that are being used in patients affect cells in the absence of viral infection. And that project has actually led to some interesting genetic targets that we think affect how well these drugs work, and they're kind of giving us some new insights that probably not directly would be used by clinicians, but would at least kind of help the drug development process of what makes these drugs, what limits the amount of these drugs that we can give to patients because of toxicity concerns. We also have another project that's basically trying to understand the one of the genes in SARS-CoV-2 called RDRP, or RNA-dependent RNA polymerase, using cloning to develop fluorescent reporter assays. But probably without going on a long tangent, that's probably the best place to leave it.
Sure. And actually, well, staying on that topic for just a moment more, this is maybe a speculation question for you. Do you think that CRISPR-Cas9 can be helpful in finding COVID-19 accurate vaccine?
I think probably not in a direct way. There's some really interesting developments in CRISPR-Cas9 on using it as a diagnostic test. So that's more in the area of testing for SARS-CoV-2 or COVID. And there, I think if this pandemic had happened a few years later, you'd probably be seeing CRISPR-Cas9 based testing approaches as the most common ones that are being used, as opposed to right now, most of the tests that are being used are PCR based tests. But the CRISPR-Cas9 ones are actually more sensitive. It's just that the technology is so new that getting it to the point where it's validated enough to kind of be so sure of it that it has the right accuracy was just not quite there yet. But I think I think also just the knowledge of how important it is to have readily available and quick turnaround tests, I think you'll see a lot of development in that area with CRISPR-Cas9. In terms of the therapeutics, I think the pipelines for developing vaccines are pretty well established, and as far as I know, do not use CRISPR-Cas9. Certainly, they would use molecular cloning. There's a lot of basically you want to clone the antigens that you're going to use, the peptides or different regions of the SARS-CoV-2 genome that you use to develop vaccines or antibodies. There's a lot of use in cloning those. I think CRISPR-Cas9 as a therapy is more like 10 to 20 years off. I mean, as a therapy for any genetic disease, it's only really starting to be used in trials to the point where it could be used as an effective therapy against a virus. If it ever gets there, it would be 10 to 20 years away.
All right. A different type of technical question here. Why is there a limitation in the addition or deletion of just 44 base pairs?
It has to do with the way that this prime editing technology works. That it basically works through what you're actually doing is you have the Cas9 enzyme, which directs the Cas9 to a place in the genome, and actually attached to Cas9 you have a reverse transcriptase enzyme that actually reverse transcribes a sequence hanging off of your guide RNA and primes that off of the genome. So you're actually directly extending a nicked single-strand break site in your genome with a sequence that you've attached onto your CRISPR guide RNA. And I guess just at some point when you have too long of an RNA sequence attached to your guide RNA, it stops functioning properly, and that's really where this limitation currently comes from. But the paper discussing or revealing this CRISPR prime editing technique is 6 months old. So I think in the next year or two, probably those limitations will change.
Great. This is such an interesting question. It was prefaced as a silly question, but I think it's great. What happens to all the materials that are used after these experiments are completed? Is there any kind of a recycling process?
So I guess there's two versions of materials. There's the biological materials, like any plasmid that we've cloned. We store them because you never know when you're going to use things again. Especially with these CRISPR guide RNAs, one project might target a specific gene, and then maybe 3 years later, someone else's project might want to target the same gene for a different reason. So the plasmids we tend to store. There's a lot of other reagents that go into this process, plastics, purification reagents. Those are all disposable, and certainly labs in the Harvard area care a lot about this idea of, can you make things more sustainable? However, I think more important than that is that with plasmids, one copy of a plasmid in the right bacteria can grow to be, 1 trillion copies. And so any sort of contamination is something that we worry about a lot, and happens in our lab, where we think we're cloning a particular plasmid, but then we find that, at some frequency, we've actually cloned a plasmid that was just kind of, sitting around in a pipette tip from a year ago. So I do think that there's a lot to be said for making research process sustainable, but also without compromising on the idea that disposability allows us to not to get contaminations.
Thank you. And I think we could keep you here all day asking questions, but I'll limit it to 2 more. And we appreciate your generosity with your time. One is, how is CRISPR-Cas9 used in stem cell technology?
Yeah, so there's a lot of interest in the stem cell field in modeling diseases or modeling possible cures diseases, because one of the amazing things about stem cells is the ability to turn stem cells into a cell type that could be useful in disease modeling or potentially transplantation. And that's, for example, one great example is, it's very hard to study human neuropsychiatric disorders like schizophrenia, Alzheimer's, Lou Gehrig's disease, because you can't get human brain cells to study in a dish. Whereas with stem cells, you can actually just turn a stem cell from any genetic background into that type of cell. And so one of the really exciting uses of CRISPR is making those genetic changes that will model one of these neurodevelopmental disorders, for example, or potentially asking, are there genetic things that we could do to actually cure that disease? Not only in stem cells, but also there's now actually been patients who had hemophilia or sickle cell anemia, basically these blood disorders, where you can actually use CRISPR to correct the mutation in a blood stem cell, transplant that cell into a patient, and it will actually correct the genetic disorder that they have and basically repopulate their blood with healthy cells.
Great, thank you. And I know we have more exciting questions coming in the chat, so perhaps if you have a few moments, feel free to take a look at those. But the last question I'd like to put to you, what are the political and social constraints that your lab faces during cloning known and foreign genes with the technology that you're using?
Yeah, that's a really good question. I mean, the only case I can think of is when we order a large oligonucleotide, or basically a gene to be that we're cloning, we have to, on an honor system, basically report that it is not a known toxin. That's the only case I can think of where, and then obviously the NIH has rules and regulations. We have to abide by the safety standards of the NIH that basically involve the same thing. We can't, create toxins. We can't mess with viruses, with infectious viruses. When we're talking about this lentivirus system, which is really the only viral platform we use, there's many layers of safety that prevent it from replicating inside cells. Same thing, we have a collaboration that's studying COVID-19, or where we, because of this project, we actually wanted to do an experiment that involved infecting cells with infectious SARS-CoV-2. That's done in this biosafety level 4 setup at Boston University, where there's an incredible amount of precautions made to prevent this from getting outside of the biocontainment. So I guess from a safety perspective, I think that there's a lot, again, with the idea that researchers rely on federal funding. And so if you put these kind of checks and balances on federal funding, it tends to be an effective way of regulating what scientists do. The ethical issues, I guess, kind of that relates more to these ideas of, with CRISPR-Cas9 especially, is there a line between curing a disease and improving, basically creating a particular sort of genetic change that would amount to improving a human? For example, a commonly used example is changing the eye color. That's a genetic change that would be pretty easy to do, but would it be ethical to make that sort of change? And I think that could spur a whole conversation. But as a one sentence answer, honestly, I think a lot of the policing goes on at the level of us as scientists, making, talking about experiments we do, making sure that there are ethical standards. And of course, our research is all at the basic level, cells in a dish. Once you start doing things that would go into patients, there are institutional review boards, there are FDA checks. So I think there's a whole level of checkpoints of what qualifies as ethical once it's going into a human patient.
Wonderful. Well, thank you so much, Dr. Sherwood. There's many expressions of thanks and appreciation for your lecture today. So thank you so much for joining us. It is so foreign not to be able to applaud for you at the end of a talk. So I'm not sure what the Zoom equivalent is, but thank you again for your time today. It was a pleasure having you.
No problem.